Tonight's lecture is over Chapter 7, Biotechnology. And our learning objectives for this chapter are to describe the basic tools used in the application of biotechnology, to describe what CRISPR is, and the steps for how it is used in biotechnology. Also, we should be able to describe the potential benefits and risks with genetically modified foods. We'll be able to explain how biotechnology can help produce pharmaceuticals, treat disease, and even prevent disease. We'll be able to define such terms as genetic engineering, recombinant DNA technology, PCR, and transgenic organisms. We will also be able to identify the strengths and weaknesses of gene therapy. We will be able to describe the steps used in cloning an organism, and we will be able to describe how to create a DNA fingerprint. Organisms, cells, and their molecules can be modified and engineered to achieve practical benefits for humans. Researchers who participate in this endeavor are part of the field known as biotechnology, which has had some remarkable successes and generated exciting possibilities, but it also has some potential pitfalls. When you hear about biotechnology in the media, it is important to be cautious in distinguishing successes from promise. You've probably seen intriguing advertisements for products that promise insights into specific genes you carry and what they have to say about your health and ancestry. Or you may have read popular media stories highlighting attention-grabbing promises of biotechnological research such as the following: a kit that can be used as a test for cancer with just one drop of saliva, a device that can stimulate regeneration of neurons following spinal injuries, targeted delivery of genetically engineered cells that can cure inherited diseases, and quote-unquote gene chips that can identify your risk of developing any one of hundreds of diseases and highlight treatments personalized for your genome. As of yet, none of these promises has come to fruition. Meanwhile, most scientists are conservative when interpreting their results and cautious when drawing conclusions or when generalizing from limited observations to broader assertions about their significance. Reconciling the overblown promises that appear in the media and the much more conservative conclusion of scientists when discussing biotechnology can be difficult. But realize that scientific methods are often used in the pursuit of practical applications, and in such cases, economic considerations can become important. As entrepreneurs seek to establish the value of the products that they are developing, they may court the media with reports highlighting the potential of their products, which may have been demonstrated only in cell cultures, animal models, or extremely limited numbers of cases in humans. And oftentimes, the popular media get caught up in the excitement and conflate the promise and unsubstantiated claims with reality. Let's look at an example. In 2019, researchers at a biotech company announced that they could offer a complete cure for cancer in a year's time by using a combination of cancer-targeting peptides and a toxin that specifically kills cancer cells. Their sweeping claims were repeated in more than 700 news articles, including glowing stories in Forbes and the New York Post that were viewed by more than 2 million people. These articles were shared widely across social media, including nearly 4 million times on Facebook. The researchers' claims, however, were based on just a few unpublished experiments on cells grown in petri dishes and one in mice. None were reviewed by outside scientists. Within days, cancer research experts around the world began weighing in, pointing out the complete lack of any evidence supporting the claims. No data from any controlled studies were provided, nor was a single trial conducted in humans. The virtually unanimous verdict from the outside experts and cancer organizations was that the unsubstantiated claims were irresponsible and cruel and almost certainly untrue. Subsequent fact-checked articles correcting the initial sensational claims and including evaluations by relevant experts were viewed and shared much less frequently. As a result, many people were left with erroneous and potentially dangerous beliefs. There is nothing unusual about these studies. They simply reflect a common process by which unsubstantiated and often untrue information is spread. Hearing the report in the media, however, people might reasonably but erroneously have assumed that this was a victory for biotechnology. Moving on, this is not to say that there have not been major victories in biotechnology. The steadfast work of biotech researchers has yielded successes that are robust and are changing our world in significant ways. The achievements made in agriculture, human health, and forensic science are particularly notable. For instance, in agriculture, the development of crops resistant to pests and disease has increased yields dramatically while reducing costs and, in many cases, reducing the amount of pesticides that must be used. Additionally, biotechnology can be used to produce foods with enhanced nutrition. Also, human health. Biotechnology has led to some notable successes in treating diseases and to improvements in diagnosing and screening for genetic diseases. Also, forensic science. Forensic biotechnologies have led to spectacular advances in the capability of law enforcement and improvements in the criminal justice system. And perhaps equally important, the lessons we're learning from these sobering cases are helping us to reform the criminal justice system. Any discussion of biotechnology must acknowledge that alongside the potential benefits come difficult ethical issues, potentially harmful environmental impacts, and possible health risks. These must be addressed at every stage. And that brings us to our take-home message for Section 7.1. Biotechnology is the use of technology to modify organisms, cells, and their molecules to achieve practical benefits primarily in agriculture, human health, and forensic science. Moving on, some common technologies, techniques, or tools are employed in the applications of biotechnology. Moving on, how would you create a plant resistant to being eaten by insects, or a colony of bacteria that can produce human insulin, or microbes to convert waste frying oils into biodiesel fuel? Surprisingly, although there are many different applications of biotechnology, only a relatively small number of processes and tools are used. These enable researchers to chop up the DNA from a donor organism that exhibits the trait of interest, amplify the small amount of DNA into larger quantities, insert pieces of the DNA into bacterial cells or viruses, then grow separate colonies of the bacteria or viruses, each of which contain a different inserted piece of donor DNA. So first, in chopping the DNA, researchers start by selecting a donor organism that has a desirable trait. Suppose they want to produce human growth hormone in large amounts. The first step would be to obtain a sample of human DNA and cut it into smaller pieces using restriction enzymes. Restriction enzymes evolved in bacteria to defend against attacks by viruses, and their job is to cut DNA into small pieces. Dozens of different restriction enzymes exist, each of which recognizes and cuts a different specific sequence of bases in the DNA. Then we must amplify the DNA. In biotechnology, it is common for only a small sample of the DNA of interest to be available. For this reason, the polymerase chain reaction, or PCR, which was invented by Kary Mullis, is a valuable tool for researchers. PCR is a laboratory technique that allows a tiny piece of DNA to be duplicated repeatedly. The process involves briefly heating a solution containing the DNA of interest, which causes the two strands of DNA to separate, or we would call that melt. As the solution cools, an enzyme covalently bonds free nucleotides with their complementary bases on each of the separated single strands of DNA. Adding primers with sequences complementary to the region that is desired to be amplified makes it possible to selectively amplify regions of interest. The result is two complete double-stranded copies of the DNA, and the process can be repeated again and again until there are billions of identical copies of the original sequence. And that brings us to inserting the DNA. So to insert foreign DNA into a target organism, a process known as gene editing or genome editing alters an organism's genome. In the human growth hormone, for example, researchers might want to transfer the human growth hormone gene into the bacterium E. coli, creating transgenic organisms, that is, organisms with DNA inserted from a different species. The bacteria can then produce large amounts of the desired product. To create a transgenic organism, researchers must physically deliver the DNA from a donor species into the recipient organism. This delivery requires a vector. A vector is something to carry the donor DNA, and it is often accompanied by using plasmids, which are circular pieces of DNA that can be incorporated into a bacterium's genome. So remember, I talked about plasmids in a previous chapter. A restriction enzyme then recognizes and binds to a particular sequence of 4 to 8 bases of the plasmid and cuts there, thus allowing insertion of the matching sequence of donor DNA, matching because it was cut by the same restriction enzyme. After the insertion of the plasmid into the bacterial cell, genes on the plasmid can be expressed in the bacterium and are replicated whenever the cell divides. So both of the new cells contain the plasmid with the donor gene. In other cases, genes are incorporated into viruses instead of plasmids. The viruses can then be used to infect organisms and transfer the genes of interest into those organisms. And that brings us to growth. Once a piece of foreign DNA has been transferred to a bacterial cell, every time the bacterium divides, it creates a clone, a genetically identical cell that contains the inserted DNA. With numerous rounds of cell division, it is possible to produce a huge number of clones, all of which transcribe and translate the gene of interest. In a typical recombinant DNA experiment, that is, one in which DNA from 2 or more sources is brought together, a large amount of DNA may be chopped up with restriction enzymes, incorporated into the plasmids, and introduced into bacterial cells. The bacteria are then allowed to divide repeatedly, with each bacterial cell producing a clone of the foreign DNA fragment it carries. Together, all of the different cells containing all of the different fragments of the original DNA are called a clone library or a gene library. Researchers can later identify those bacteria with the gene of interest and grow them in large numbers. And that brings us to our take-home message for section 7.2. Methods including the use of naturally occurring restriction enzymes for cutting DNA, the polymerase chain reaction, or PCR, for amplifying DNA, insertion of DNA into vectors, and cloning make it possible to cut and copy DNA from one organism and deliver it into another organism. Moving on, since 2012, one technology has generated almost unprecedented enthusiasm while racking up some impressive successes in delivering on its early promise. It goes by the acronym CRISPR, is pronounced CRISPR, and it is a system for editing DNA. What makes CRISPR noteworthy is that it brings much greater precision and efficiency to gene editing, acting as a cut and paste tool that enables researchers to modify almost any gene in any organism. CRISPR stands for Clustered Regularly Interspersed Short Palindromic Repeats. The name describes the organization of DNA that originally came from viruses but regularly is incorporated by bacteria within their own genomes to serve as a sort of immune system that can help thwart infection by potential lethal viruses. Humans often develop immunity to illness-causing microbes after an initial encounter. For example, if you contract measles as a child, you won't contract it later as an adult. Likewise, exposure through vaccination protects most people from contracting the disease later in life. Similarly, when a virus first infects a bacterium, the bacterium keeps a kind of record of the encounter. The bacterium copies some of the virus's DNA and integrates it into its own DNA. When the same type of virus invades again, the bacterium uses the stored virus DNA to quickly produce an RNA molecule that acts as a sort of homing device, finding and binding to the infecting virus's DNA. After the RNA latches onto the viral DNA, the bacterium uses a DNA cutting enzyme called Cas9 to cut the foreign DNA into pieces, which renders the invader harmless. This method, which enables bacteria to effectively remember and resist viral infections, turns out to be present in almost half of all bacteria. An important intellectual leap researchers made was to recognize that the useful biotechnology value of the CRISPR-Cas9 system. With its programmable RNA molecule capable of guiding the Cas9 DNA scissors to a specific sequence, the system could serve as an efficient system by which DNA could be easily and accurately edited. Building on this insight, adapting the CRISPR system for use in biotechnology is straightforward. First, after identifying a particular DNA sequence of interest, perhaps to fix or inactivate a defective gene, researchers synthesize an RNA molecule, the guide molecule, with a sequence that matches the target gene to be sliced, following the complementary base pairing rules we described in a previous chapter. The DNA sequences for CRISPR RNA and the Cas9 enzyme are introduced into the target cells using a plasmid as a vector. Thirdly, within the target or host cell, the RNA leads Cas9 to exactly the desired location on the cell's DNA, and the enzyme cuts the DNA at that point. Fourth, at that location, a sequence can then be inserted that repairs or alters in some other way the host cell's DNA. CRISPR is considered a breakthrough in biotechnology because the ability to target and snip DNA so precisely at specific sequences opens the door to changing an organism's genes in almost any way imaginable. Not only is the CRISPR system remarkably precise, but it is also fast and relatively inexpensive. Just a few of the initial successful uses of CRISPR reveal the wide variety of potential applications. For instance, increasing significantly the muscle mass in the legs of a dog, or reducing the severity of genetic deafness in mice, or removal of a gene from an embryo that causes a type of heart disease, or inactivating the immune system markers on a tissue from a pig by editing 62 genes simultaneously in a way that could facilitate the use of transplants from pigs into humans, or introducing mutations into human stem cells to produce tissues with disease properties that can serve as model systems for studying common human diseases. There is even more enthusiasm surrounding CRISPR's potential for fighting some of the most harmful diseases that affect humans. Significant work has already been done with CRISPR in altering the biochemistry of mosquitoes so that they cannot host or transmit the parasite that causes malaria. Eradicating malaria could save 500,000 lives every year. With CRISPR, it might also be possible to inactivate genes in disease-causing bacteria that confer resistance to antibiotics. Likewise, scientists may be able to target invasive species that damage environments and threaten native species. So without question, CRISPR has enormous potential, but some concerns remain beyond the legal proceedings as several universities and companies fight over who has rights to develop and profit from these technologies. In several recent studies, for example, DNA damage in CRISPR-edited cells has led to the loss of cancer-fighting mechanisms, potentially increasing the risk of tumor development. Perhaps the most significant concerns are ethical issues related to possible applications of CRISPR. These include concerns about the potential for editing human embryos or germline cells, sperm and egg cells, because the gene changes could be passed to subsequent generations. Such changes might be difficult to control and could have significant and unexpected health consequences. Critics have also noted that the consequences of introducing new or altered genes into the genomes of natural populations of organisms are difficult to predict. Even some desirable outcomes, eradicating the mosquito populations that transmit malaria, for example, might have secondary harmful effects, such as for birds or bat populations that rely on mosquitoes as a food source. And that brings us to our take-home message for section 7.3. CRISPR is a precise and efficient gene editing tool that targets and snips DNA of almost any species at a specific sequence, opening the door to changing an organism's genes in almost any way imaginable. Moving on, your breakfast cereal is probably fortified with vitamins and minerals, and when snacking, you may eat protein bars that have as much protein as a full chicken breast. So it shouldn't come as a surprise to learn that farmers use biotechnology to improve on the natural levels of vitamins, minerals, and other nutrients in fruits, vegetables, and the livestock that they produce. For thousands of years, humans have been practicing a relatively crude and slow form of genetic engineering, the manipulation of a species' genome in ways that do not normally occur in nature. In Darwinian terms, we would call this artificial selection. In its simplest form, genetic engineering is the careful selection of the plants or animals to be used as the breeders for a crop or animal population. Through this process, farmers and ranchers have produced meatier turkeys, seedless watermelons, and big, juicy corn kernels. But what previously took many generations of breeding can now be accomplished in a fraction of the time using recombinant DNA technology, the combination of DNA from two or more sources into a product. In crop plants, for example, the process begins with the identification of a desirable characteristic, such as larger size or faster ripening time. Traditionally, breeders would then search for an organism of the same species that had the desirable trait, breed it with their crop organisms, and hope that the offspring would express the trait in the desired way. With recombinant DNA technology, the desired trait can come from any species, so the pool of organisms from which the trait can be taken becomes much larger. Organisms produced with recombinant DNA technology are referred to as genetically modified organisms, or GMOs. Although genetic engineering is difficult in practice, the results so far hint at a fruitful marriage of agriculture and technology. Here are some success stories. The nutrient-rich golden rice. Almost 10% of the world's population suffers from vitamin A deficiency, which causes blindness in an estimated 250,000 children each year and a host of other illnesses. These nutritional problems are especially severe in southern Asia and sub-Saharan Africa, where rice is a staple of most diets. Researchers have developed what may be the model for solving problems with biotechnology. It involves the creation of a new crop called golden rice. Humans and other mammals generally make vitamin A from beta-carotene, a substance found in abundance in most plants. It's what makes carrots orange, but not in the edible part of rice grains. To introduce beta-carotene into rice crops, researchers used recombinant DNA technology to insert 3 genes into the rice genome that code for the enzymes used in the production of beta-carotene. 2 of the transplanted genes were from the daffodil plant, and 1 was from a bacterial gene. As the transplanted genes are expressed, the rice grain takes a golden color from the accumulated beta-carotene. Since golden rice was first developed in 1999, new lines have been created that produce almost 25 times the vitamin A found in the original strains. Field tests of golden rice are still in progress, but it is viewed as one of the most promising applications yet of biotechnology. Precautionary biosafety protocols and regulations, however, have restricted its cultivation by large-scale commercial farms. As of 2019, 154 Nobel laureates have signed a letter urging an end to the opposition to GMOs, highlighting the potential value of golden rice in particular. And that brings us to insect and herbicide resistance. In the United States, biotechnology has already had a profound impact on agricultural practices. It is not a stretch to say that we are in the midst of another green revolution, but that few people are aware of it. The numbers are surprising. 86% of all corn grown in the United States is genetically modified. 93% of all cotton grown is genetically modified, and 93% of all soybeans grown are genetically modified. Two factors largely explain this extensive adoption of genetically modified plants in U.S. agriculture. Firstly, many plants have insecticides engineered into them, which can reduce the amounts of insecticide that must be added during agriculture. Second, many plants also have herbicide-resistant genes engineered into them. The cultivation of herbicide-resistant plants can reduce the amount of plowing needed to remove weeds. As a consequence, the use of genetically modified plants can reduce both the cost of producing food and the loss of topsoil to erosion. Every year, about 40 million tons of corn is deemed unmarketable as a consequence of insect damage. Increasingly, however, farmers have been enjoying greater success in their battles against insect pests. Farmers owe much of this success to a soil-dwelling bacteria of the species Bacillus thuringiensis, or BT, which produce spores containing crystals that are fatally poisonous to insects but harmless to the crop plants and to people. Beginning in 1961, the toxic BT crystals were included in the pesticides sprayed on crop plants. Then in 1995, the gene coding for production of the BT crystals was inserted directly into the DNA of many crop plants, including corn, cotton, and potatoes. Because the plants themselves produced insect-killing BT crystals, farmers no longer needed to apply BT-containing pesticides. There is no evidence that humans are harmed by the BT crystals, even when they are exposed to very high levels. In the fight against weeds, bacteria have proven very useful as well. In the 1990s, researchers discovered a bacterial gene that confers resistance to herbicides. Integration of this gene into the plant's DNA gives the crops resistance to herbicides, allowing farmers to kill weeds with herbicides while leaving the crop plants unharmed. Agriculture includes the cultivation of not just plants, but also animals. In 2015, the U.S. Food and Drug Administration, the FDA, for the first time approved for human consumption a genetically modified animal, a transgenic Atlantic salmon. The salmon carries a growth hormone from another species, the Chinook salmon, as well as a region of DNA from a third species, the ocean pout, that acts on a switch, facilitating transcription of the growth hormone gene. The transgenic Atlantic salmon, which is reported by its creators to taste the same as regular Atlantic salmon, grows much more quickly and reaches market size within 18 months rather than the usual 3 years. According to the FDA, transgenic salmon is quote-unquote as safe to eat as food from other Atlantic salmon and is as nutritious. As of 2019, the FDA indicated that the USDA's National Bioengineered Food Disclosure Standard has been satisfied, allowing the sale of the genetically modified salmon. As of right now, March of 2024, such fish have not yet come to the market. And some fisheries experts, food safety experts, environmental groups, and consumer groups have expressed apprehension at eating the salmon that they might cause increased rates of allergic reactions or have other adverse effects. There is also the fear that transgenic fish will escape from their enclosed breeding facilities into the natural habitat. If this occurred, transgenic fish might outcompete wild populations for resources and grow too large to be consumed by natural predators. The wild salmon populations, many of which are listed as endangered, could suffer as a result. And that brings us to our take-home message for section 7.4. Biotechnology has led to improvements in agriculture through the use of transgenic plants and animals to produce more nutritious food. Biotechnology has also reduced the environmental and financial costs of producing food through the creation of herbicide-resistant and insect-resistant crops. The potential ecological and health risks of such widespread use of transgenic species are not fully understood. Moving on, chicken without feathers may look ridiculous. Nevertheless, a genetically modified breed of featherless chicken was developed to be easier and less expensive to prepare for the market, benefiting farmers by lowering their cost and benefiting consumers by lowering prices. These quote-unquote naked chickens, however, have turned out to be unusually vulnerable to mosquito attacks, parasites, and disease, and are ultra-sensitive to sunlight. They also have difficulty mating, owing to the male's inability to flap their wings. Naked chickens teach us an important lesson about genetically modified plants and animals. Although the breed of featherless chickens was produced by relatively low-tech genetic engineering, the traditional animal husbandry method of crossbreeding, the desired trait of no feathers was accompanied by unintended and undesirable traits. Now, as more genetically modified foods are produced with modern methods of recombinant DNA technology, we must evaluate the risk of creating unintended and potentially harmful traits. For these and other reasons, some legitimate and rational, others irrational, many people have concerns about the production and consumption of genetically modified foods. Well, let's talk about concerns regarding genetically modified foods. One is the safety of eating genetically modified foods. The concern about the safety of eating genetically modified foods was also addressed in 2016 in a report by the National Academy of Sciences and an independent organization of the most distinguished experts in science. In this careful and detailed 606-page report, the National Academy of Sciences examined epidemiological data on cancer rates and other human health problems and concluded that there was no evidence that foods from genetically engineered crops were less safe than those from non-genetically engineered crops. The National Academy of Sciences also noted that a large number of experimental studies provided reasonable evidence that animals, too, were not harmed by eating food derived from genetically modified crops. My response to this is that we still don't really know over the long term if these GMOs will cause harm. This technology is very young, and while we may be able to do some short-term experiments, we can't really see that in the long term. So there's another concern about GMOs, and that is the invincibility of organisms that we want to kill. Herbicide-resistant canola plants were cultivated in Canada, making it possible for farmers to apply herbicides freely to kill the weeds but not the canola crop. But the herbicide-resistant canola plants accidentally spread to neighboring farms and grew out of control. Because traditional herbicides could not kill them, similarly, there has been concern that insect pests will develop resistance to the Bt produced by genetically modified crops, which will also make the pest resistance to Bt pesticides applied to crops that are not genetically modified. Then in a National Academy of Sciences report, scientists noted that insects have been slow to evolve resistance and resistance management strategies have been effective. Overall, they found no conclusive evidence of cause and effect relationships between genetically engineered crops and environmental problems. Well, I think the key word there is slow to evolve resistance. They're still evolving resistance. So again, we have to be really careful with this technology. And there's yet another concern about GMOs, and that is the adequate testing and regulation of the GMOs. It is impossible to really know whether a new technology has been tested adequately. Scientists and lawmakers have been working toward a responsible set of policies designed to ensure that sufficient safety testing is done. For example, laboratory procedures for working with recombinant DNA have been established, and researchers have developed techniques that make it impossible for most genetically engineered organisms to survive outside the specific conditions for which they are developed. Additionally, regulatory strategies vary from country to country. And he gave it for his opinion that whoever could make 2 ears of corn or 2 blades of grass to grow upon a spot of ground where only one grew before would deserve better of mankind and do more essential service to his country than the whole race of politicians put together. Jonathan Swift, Gulliver's Travels, 1726. So I think that it's good that there are standards in place for the testing and regulation of GMOs. However, the test of time will tell. I know that GMOs have helped drive the organic food market. There is another concern about the loss of genetic diversity among crop plants. As increasing numbers of farmers favor one or a few genetically modified strains of crops, the genetic diversity of crops declines. This can increase the vulnerability of crops to environmental changes or pests. For example, in the mid-1800s, much of the population of Ireland depended on a diet of potatoes. Because most of the potato crops had been propagated from cuttings from the same plant, they were all genetically the same. When the crops were infected by a mold specific to that one crop, most were wiped out, causing the Irish potato famine, which was ultimately responsible for the deaths of more than 1 million people. Another concern is the hidden costs of GMOs. When seed companies create genetically modified seeds with crop traits desirable to farmers, the companies also engineer sterility into the seeds. As a consequence, the farmers are dependent on seed companies and must purchase new seeds for each generation of their crops. In most situations, however, seed costs are only a small fraction of the total costs of crop production. Yeah, well, a small fraction is still a fraction, and I can tell you as a small business owner myself that sometimes little costs, they add up to big costs. Another concern is that unnatural substances are necessarily harmful. Some argue that genetically modified foods are not natural and for that reason must be harmful. This argument is called the naturalistic fallacy. It is a common but flawed argument and should not be cause for concern. Smallpox, HIV, poison ivy, and cyanide are all natural and obviously harmful to humans. The smallpox vaccine, on the other hand, is unnatural and obviously helpful to humans. Innumerable other valuable technological developments are equally unnatural. In most cases, there simply is no value in knowing whether something is natural or unnatural when evaluating whether it is good and desirable. Well, my response to that is I think that it's equally fallacious to say there is no value in knowing whether something is natural or unnatural. So to make an informed judgment regarding GMO foods, we must compare the risks of producing genetically modified foods with the benefits. These analyses should include the potential to increase food production and reduce food costs, the ability to reduce environmental degradation by agriculture, and the safety of agricultural workers. For example, with genetically modified, pest-resistant crops, farm workers will greatly benefit from spending less time applying pesticides. This is particularly important for workers in less developed countries, where safety regulations for pesticides are often ignored or unenforced. Unfortunately, establishing the environmental risks of genetically modified foods is difficult. This important issue must be evaluated closely. And that brings us to our take-home message for section 7.5. More genetically modified foods are being created through modern methods of recombinant DNA technology. For the public, however, some fears remain about the potential risks of these foods and about the long-term financial advantages they offer. Moving on, in the best of all possible worlds, biotechnology would prevent debilitating human diseases. The next best would be to cure diseases forever. but these are difficult, long-term goals, so biotech often is directed at the more practical goal of treating diseases, usually by producing medicines more efficiently and effectively than with traditional methods. Biotechnology has had some notable successes in achieving this goal. The treatment of diabetes is one such success story. Type 1 diabetes, often called juvenile diabetes, is a chronic disease in which the body cannot produce the hormone insulin. Without insulin, the body's cells are unable to take up and break down sugar from the blood. Type 2 diabetes, which accounts for 90% of all cases of diabetes, is a metabolic disorder characterized by elevated blood sugar levels resulting from insulin resistance and insufficient insulin production. Complications from both types of diabetes can be deadly. Approximately 1/3 of all people with diabetes treat their condition with one or more daily injections of insulin. As recently as 1980, the insulin necessary to treat diabetes was extracted from the pancreas of cattle or pigs that had been killed for meat. This process of collecting insulin was difficult, costly, and time-consuming. Everything changed in 1982 when a 29-year-old entrepreneur named Bob Swanson joined scientists Herbert Boyer and Stanley Cohen to realize the potential of recombinant DNA technology for insulin production. The team used restriction enzymes to snip out the human DNA sequence that codes for the production of insulin. They then inserted this sequence into the bacterium E. coli, creating a transgenic organism. After cloning the new transgenic bacteria, the team was able to grow vats of the bacterial cells, all of which churned out human insulin. The drug could be produced efficiently in huge quantities and made available for patients with diabetes. This was the first genetically engineered drug approved by the FDA, and it continues to help millions of people every day. This application of biotechnology revealed a generalized process for genetic engineering and, in doing so, launched the biotech revolution. It instantly reopened the door to a more effective method of producing many different medicines to treat diseases. Today, more than 1,500 companies work in the recombinant DNA technology industry, and their products generate more than $500 billion in revenue each year. Several important achievements followed the development of insulin-producing bacteria, and I'm going to illustrate just two examples for you. One is human growth hormone. Produced by the pituitary gland, human growth hormone stimulates protein synthesis, increases the use of body fat for energy to fuel metabolism, and stimulates the growth of virtually every part of the body. Insufficient growth hormone production, usually due to pituitary malfunctioning, leads to dwarfism. When treated with supplemental HGH, individuals with dwarfism experience additional growth. However, until 1994, HGH treatment was prohibitively expensive. HGH could be produced only by extracting and purifying it from the pituitary glands of human cadavers. Today, transgenic bacteria produce HGH in virtually unlimited supplies. And that brings me to erythropoietin. Produced primarily by the kidneys, erythropoietin, also known as EPO, is a hormone that regulates the production of red blood cells. Many clinical conditions, nutritional deficiencies and lung disease among others, and medical treatments such as chemotherapy can cause individuals to have a lower than normal number of red blood cells, a condition called anemia. Anemia reduces the transport of oxygen to tissues and cells, and this lack of sufficient oxygen causes a variety of symptoms, including weakness, fatigue, and shortness of breath. Recombinant human erythropoietin, first cloned in 1985, is now produced in large amounts in cells originally derived from hamster ovaries. It is used to treat many forms of anemia. Worldwide sales of EPO are in the billions of dollars. EPO has been at the center of several blood doping scandals in professional cycling. Because this hormone increases the oxygen carrying capacity of blood, some otherwise healthy athletes have used EPO to improve their athletic performance. This practice can be very dangerous, though. Increasing the number of red blood cells causes the blood to become much thicker, increasing the risk of heart attack. The list of other medicines currently produced by transgenic organisms is long. Some, such as Factor VIII, a treatment for hemophilia, generate more than $1 billion in sales per year. Beyond these, plans are underway to create a variety of other useful products for treating diseases, including potatoes that produce antibodies that enable a more effective response to treat an illness. And with animal species that are used to create medicines, we call these animals farm animals. And I'm not talking F-A-R-M, I'm talking P-H-A-R-M. Farm animals. And that brings us to our take-home message for section 7.7. Biotechnology has led to some notable successes in treating diseases, usually by producing medicines more efficiently and effectively than they can be produced with the traditional methods. Moving on, how much do you want to know about your future? If you could carry a gene that means you are likely to develop a particular disease later in life, or if there is a large chance that your children will be born with a genetic disease, would you want to know? As biotechnology develops the tools to identify some of the genetic time bombs that many of us carry, these are questions that we all must address. Screening for genetic disorders generally focuses on 3 distinct scenarios. 1. Is a given set of parents likely to produce a baby with a genetic disease? Many genetic diseases occur only if an individual inherits 2 copies of a disease-causing gene, one from each parent. This is true for Tay-Sachs disease, cystic fibrosis, and sickle cell anemia, among others. Individuals with only a single copy of the disease-causing gene never fully show the disease, but they may pass on the disease gene to their children. In these cases, it can be beneficial for potential parents to be screened to determine whether they carry a disease-causing copy of the gene. Such screening, combined with genetic counseling and testing for embryos, can dramatically reduce the incidence of a genetic disease. Recently, researchers have begun advocating genetic screening of heritable factors associated with heart disease, breast cancer, and ovarian cancer. Second, will a baby be born with a genetic disease? Once fertilization has occurred, it is possible to test an embryo or developing fetus for numerous genetic problems. Prenatal genetic screening can detect cystic fibrosis, sickle cell anemia, Down syndrome, and a rapidly growing list of other disorders. Such screening is recommended if a woman has had positive results from a prenatal screening test, typically based on having risk factors, has had a chromosomal condition in a previous pregnancy, is age 35 or older, or has a family history of a specific genetic condition, or if the woman or her partner is a known carrier of a genetic condition. To screen the fetus, doctors must examine some of the fetal cells and or the amniotic fluid, that is the fluid that surrounds the fetus in the uterus and contains many chemicals produced by the developing embryo. Cells and fluid are usually collected by amniocentesis or chronic villus sampling techniques that we explore in detail in a subsequent chapter. Increasingly, these cell and fluid collection methods are being replaced by non-invasive methods requiring only a blood sample from the mother. And the third question is an individual likely to develop a genetic disease later in life? DNA technology can be used to detect disease-causing genes in individuals who are currently healthy but are at an increased risk of developing an illness later. Early detection of many diseases such as breast cancer, prostate cancer, and skin cancer greatly enhances the ability to treat a disease and reduce the risk of more severe illness or death. These potential benefits of genetic technology also come with potential costs. People who have a gene that puts them at increased risk of developing a particular disease, for example, might face discrimination from employers or insurance companies, even though they are not currently sick and may never suffer from that particular disease. Although the Genetic Information Non-Discrimination Act was signed into law in the United States in 2008, it does not cover discrimination by life insurance, disability insurance, and long-term care insurance companies. In fact, some insurance companies have already denied coverage when they discover that an individual carries a gene that puts him or her at increased risk of disease. Another problem is that parents who discover that their developing fetus will develop a painful, debilitating, or fatal disease soon after birth are confronted with difficult questions about how to proceed. When it comes to curing a disease by using biotechnology, there is a small glimmer of hope. Since the 1990s, a small number of humans with a usually fatal genetic disease called severe combined immunodeficiency disease, or SCID, SCID, have been completely cured through the application of biotechnology. Unfortunately, it has not been possible to apply these promising techniques to other diseases. And it's not for a lack of trying. More than 2,600 clinical trials have focused on gene therapies designed to treat or cure a variety of diseases by inserting a functional gene into an individual's cells to replace a defective version of the gene. But so far, there have been very few clear successes. Well, let's look at what's been accomplished in treating SCID, a condition in which a baby is born with an immune system unable to properly produce a type of white blood cell. The infant is vulnerable to most infections and usually dies before his or her first birthday. In gene therapy for SCID, researchers remove stem cells from an affected baby's bone marrow. These stem cells have the ability to develop into any type of cell in the body. In the bone marrow, they normally produce white blood cells, but in individuals with SCID, a malfunctioning gene disrupts normal white blood cell production. Next, in a test tube, the infant's bone marrow stem cells are infected with the transgenic virus carrying the functioning gene. The virus inserts the good gene into the DNA of the stem cells, which are then injected back into the baby's bone marrow. There, the cells can produce normal white blood cells, permanently curing the disease. Several dozen cases of SCID have been cured, but 2 patients died from the illness related to their treatment. As a result of advances, however, a variant of this treatment has been approved in Europe. While no gene therapy treatments have been approved in the United States as of 2019, researchers are hopeful that the situation will change in the coming years. The most likely treatments are for hemophilia B, sickle cell anemia, and a brain disease called cerebral adrenoleukodystrophy. Difficulties in gene therapy usually relate to the transfer of the normal functioning gene into the cells of a person with the genetic disease. 1. Difficulty getting the working gene into the specific cells where it is needed. 2. Difficulty getting the working gene into enough cells and at the right rate to produce a physiological effect. 3. Problems arising from the transferred gene getting into unintended cells. And 4. Challenges in regulating gene expression. Beyond these technical problems, some of which may benefit from the CRISPR system described in an earlier section, the malfunctioning gene has not been identified for most diseases. In other cases, the disease is caused by more than one malfunctioning gene. Additionally, it is important to keep in mind that gene therapy targets cells in the body other than the sperm and the egg. Consequently, while a disease might, in theory, be cured in the individual receiving the therapy, he or she can still pass on the disease-causing genes to offspring. It's not clear what the future holds for gene therapy, but a great deal of research is in progress. And that brings us to our take-home message for section 7.8. Gene therapy has had limited success in curing human diseases, primarily because of technical difficulties in transferring normal functioning genes into the cells of a person with a genetic disease. Moving on, cloning, perhaps no scientific word is as emotionally loaded. Let's clarify what the word means. Cloning refers to a variety of techniques, including the creation of new individuals that have exactly the same genome as the donor individual, a process called whole organism cloning. The clone is like an identical twin, except that it may differ in age by years or even decades. It is also possible to clone tissues such as skin and entire organs from an individual's cells. Cloning took center stage in the public imagination in 1997 when Ian Wilmot, a British scientist, and his colleagues announced that they had cloned a sheep. They named this sheep Dolly. Their research was based on ideas dating back to 1938 when Hans Spemann first proposed the experiment of removing the nucleus from an unfertilized egg and replacing it with the nucleus from a cell of a different individual. The process used by Wilmot and his research group was difficult and inefficient, but also simple in concept. They removed the nucleus from a mammary gland cell of a grown sheep and inserted it into the egg of another sheep from which the nucleus had been removed. The egg was induced to divide as if it were a naturally fertilized egg, and they transplanted it into the uterus of a surrogate mother sheep. Out of 272 tries, they achieved one success, but that was enough to show that cloning was possible from an adult animal. Shortly after the news of Dolly's birth, teams set about cloning a variety of other species, including mice, cows, pigs, and cats. Not all of this work was driven by simple curiosity. For farmers, cloning could be an efficient method of producing animals with desirable traits, such as increased milk production in cows. Medical researchers, too, see much gain from cloning. In particular, transgenic animals containing human genes, such as the hamster ovary cells producing the RHU-EPO discussed earlier in this chapter, can be very valuable. But can a human be cloned? At this point, it is almost certain that the cloning of a human will be possible. But it is nearly unanimous among scientists that human cloning to produce children should not be attempted. Some of the reasons cited relate to the problems of the safety for the mother and the child, legal and philosophical issues relating to the inability of cloned individuals to give consent, problems of the exploitation of women, and concerns regarding the identity and individuality of the clone. Governments are struggling to develop wise regulations for this new world reality. And that brings us to our take-home message for section 7.9. Cloning of individuals has potential benefits in agriculture and medicine, but ethical questions will linger. Moving on, in another time, Colin Pitchfork, a murderer and rapist, would have walked free. But in 1987, he was captured and convicted thanks to his DNA and is now serving 2 life sentences in prison. Pitchfork raped and murdered 2 15-year-old high school girls in a small village in England in the 1980s. The police thought that they had caught the perpetrator when a man confessed, but only to the second murder. He denied any involvement in the first murder, which perplexed the police because the details of the 2 crimes strongly suggested a single culprit. Around the same time, British biologist Alec Jeffreys made an important discovery. Certain small stretches of DNA in human chromosomes are tremendously variable in their base sequences. In much the same way that each person has a driver's license number or a social security number that differs from everyone else's, it is extremely unlikely that 2 people would have identical sequences at these locations. Thus, investigators could compare the suspect's DNA sample against the DNA containing evidence left at the crime scene to identify the perpetrator. Jeffries analyzed DNA left by the murderer-rapist on the two victims and found that it did indeed come from a single person, and that person was not the man who had confessed to one of the two crimes. The suspect was released and has the distinction of being the first person cleared of a crime through DNA fingerprinting. To track down the true criminal, police collected and analyzed more than 5,000 blood samples from all men in the area of the crimes who were between 18 and 35 years old, a practice that many viewed as an invasion of privacy. This procedure led them to Colin Pitchfork, whose DNA matched perfectly the DNA left on both of the victims and ultimately was the evidence responsible for his conviction. Pitchfork almost slipped through the net, having persuaded a friend to give a blood sample in his name. But when the friend was overheard telling the story in a pub, police tracked down Pitchfork to get a blood sample. DNA fingerprinting is now used extensively in forensic investigations in much the same way that regular fingerprints have been used for a century. DNA samples are frequently left behind at crime scenes, usually in the form of semen, blood, hair, skin, or other tissue. DNA fingerprinting has been directly responsible for bringing thousands of criminals to justice and exonerating the innocent. The DNA from all humans is almost completely identical. More than 99.9% of the DNA sequences of two individuals are the same, because we're all members of the same species and thus share a common evolutionary history. Even so, in comparing two individuals' genomes of 3 billion base pairs each, a 0.1% difference still translates to about 3 million base pair differences. These differences give individuals their own unique genome. The lone exception, identical twins, whose DNA is exactly the same. Thus, the analysis of DNA from a crime scene focuses on the parts of our DNA that differ. There are thousands of these highly variable regions in the human genome. Among these thousands of variable regions, one type is used for determining a person's genetic fingerprint. These regions, called short tandem repeats, or STRs, are characterized by a short sequence, commonly 4 or 5 nucleotides long, that repeats over and over within a non-coding region of DNA. The number of repeats is what varies among individuals. So here's an example. In individual A, the number of times the sequence repeats at one STR region, say on chromosome 2, is 3 times on the maternal copy of chromosome 2 and 14 times on the parental copy. Individual A is said to have 2 different alleles for this STR region, 3 and 14. In contrast, in individual B, the same STR region on chromosome 2, the sequence repeats 5 times and 11 times. Individual B has alleles 5 and 11. For an STR region within the human genome, there are typically about 10 different alleles within a population, and each allele is shared by about 10% of all individuals in the population. So, the likelihood that 2 individuals carry the same 2 alleles is about 1 in 100. This is a low probability, but given enough people, many are likely to carry the same alleles. If 2 different STR regions are analyzed, the likelihood that 2 individuals have the same 4 alleles is 1/10 times 1/10 times 1/10 times 1/10, or 1 in 10,000. Although rarer, multiple individuals within the same metropolitan area could carry the same 4 alleles. The real power of DNA fingerprinting comes from simultaneously determining the alleles an individual carries, that is, the person's genotype, not simply at 1 or 2 STR locations, but at 13 different STR locations. This is the number used by the FBI in constructing DNA fingerprints in the United States, and it makes the probability that 2 individuals would have exactly the same genome extremely low. To produce a DNA fingerprint, an individual's genotype is determined by using PCR to amplify the STR region. Using a technique called electrophoresis, the length of the STR region is measured. The length of the region is then used to determine the number of times the STR is repeated. For a single STR region, an individual's genotype is expressed by two numbers, as described earlier, reflecting the number of STR repeats in the copies inherited from the mother and from the father. A person's full DNA fingerprint is a string of 26 numbers consisting of two numbers for each of the 13 STRs. In court, a suspect's genotype might be compared with the DNA fingerprint obtained from the evidence found at the crime scene. DNA samples from different people produce different 26-number fingerprints, whereas different samples of DNA from the same person will have exactly the same genotype. Despite universally accepted methods, DNA fingerprinting is not foolproof. Numerous incidences of human error, accidental as well as intentional, have been documented, ranging from mislabeled test tubes to tissue from a suspect being added to evidence from a crime scene. So we should not blindly draw conclusions solely from this one type of evidence. Nonetheless, DNA fingerprinting is an increasingly valuable tool for law enforcement, particularly because it is generally more reliable than eyewitness accounts. The FBI, for example, has reported that nearly 1/3 of the suspects are cleared immediately by DNA testing, and because of DNA fingerprinting, many more criminals now plead guilty to the crimes they have committed. And that brings us to our take-home message for Section 7.10. Comparisons of highly variable DNA regions can be used to identify tissue specimens and determine the individual from whom they came.